We know that we have 112 healthy individuals, astronauts in America, right? In American astronaut team, and some candidates for future space mission. And we know that International Space Station utilized only one medical so-called kit, which contained approximately 140 different medications, focused to treat more than 400 medical conditions. These conditions which expected, for example, for orbital flight, medical conditions, you can see 400 is a lot. And medical kit which presented actually here, it's very small. You can see it's small package which contained all of these pharmaceuticals, syringe, intravenous injections, many of them. But however, problem is that yes, we should treat astronauts with these pharmaceuticals. And these pharmaceuticals derived from Earth, right? They were approved by FDA, Food and Drug Administration, and released to the market with some kind of ensure people that efficacy of medication under some packaging environment will be kind of over, let's say, 1.5 years. You can take aspirin, yeah, and of course you have guarantee that 1.5 years to patency will be like 100%. By the way, FDA require that if patency or integrity of drugs, pharmaceuticals drop like 10%, it should be removed from market. Now, when we just kind of use same pharmaceuticals, which improve only for ground use, and now we start to use it in a space flight, how we know what actually consequences, whether they will be different consequences, toxic effect, pharmacology, pharmacokinetics or pharmacodynamics will be same as on the ground. Of course, we don't know this. Yeah? And more important, we know that significant alteration in physiology, like electrolytes in the blood, pressure, everything change. And this means we can expect that drug will affect probably differently than actually it was on the Earth. Second issue is that it happened that stability of drugs under space flight environment are unexpectedly less than on the Earth. And some drugs degrade in a matter of 20 days. And on this plot, you can see different pharmaceuticals, antibiotic, for example, Cipro, amoxicillin, analgetics, which actually blew before flight and after flight, 3 months flight. And you can see that they much below of FDA accepted level. This means that flight conditions affect and jeopardize integrity and potency of therapeutic potency of these drugs. Now, how we can be sure that we can treat effectively astronauts? So our laboratory, for example, and also in collaboration with Johnson Space Center, we're trying to identify what actually factors may affect on drug stability. Of course, it's radiation, humidity, temperature, vibration. By the way, vibration never was addressed actually as a very significant factor, but it's very irritating factor because if you remember, vibration mostly coming from fans. Because in absence of gravity, distribution of air is completely different. You need to push it for circulation, and these fans create vibration. And the astronauts always living under kind of noisy environment. Of course, exposure to CO2, low gravity, light, because we have over there, and the most important, repackaging. And I will address this issue later because why? Because NASA usually taking all medications and repackage them for more in some container. So as soon as they open manufacturer packaging, yeah, they jeopardize shelf life stability, right? And we need to, but it's necessary, of course, yeah. Now just we need to address very important issue whether these actually degradation of drugs in space due to only on packaging or in other factors effect. One of them, of course, radiation. We immediately can expect that, oh, in the bottom of the plot, you can see orbital flight days. And you can see in red what is increasing in milligrays dose of radiation. Of course, yeah, it's obvious to expect, yeah, longer flight, more you exposed to radiation, and radiation could be a very significant factor. At the same time, we know that some pharmaceutical companies using radiation, ionizing radiation, like gamma radiation, as a pathogen inactivation procedure. and to kill some pathogen, to ensure that some drugs is safe from infection, for example. They cannot transfer infection, especially they concern now hepatitis, for example, prion infection, which is dangerous and require a long time to appear, to have clinical actually appearance. So, but we know that yes, company has very high dose radiation to inactivate this pathway. This means that radiation probably not the factor which affect or jeopardize stability. It could be in synergetic factor to another spaceflight environment, but not alone. Like here, I present one of the repackaging consequences. Yeah, when here just a microscope and Raman spectrometry, which allow you actually to in color to identify and monitor different components of pharmaceutical formulations. And what I see, you can see here, storage on a packaging material create from the top to the bottom after 13 weeks a significant percentage of degraded material. This means that packaging itself it's a very important factor which NASA should take into account. Of course, we study drug stability. Of course, drug stability is important, physical stability, chemical stability, and microbial stability. You know, for example, in drug, we have mostly active components. Let's say analgesic ibuprofen, right? For example, but to form pill, final, you need to add a different excipients to formulate final dosage form. It's for protection, from oral, for acid in stomach, for example, if it's orally taken. It's just for better absorption, different kinds. But most important, presence of these excipients significantly affect drug stability and reduce efficacy of active pharmacological ingredients. Of course, we can see immediately how is that. We're just trying to check physical stability, for example, visual inspection. You can see change in color, cracking of some peel, or some surface change, or many kind of things which actually visually you can expect. But chemical stability is already most actually important. You cannot just kind of at one glance to say whether it's kind of there is chemical degradation or not. Why it's important? Because chemical degradation produce toxic product of degradation, which actually may cause side effects. Of course, component drug stability evaluation, which I present you just to give because we involve all in this process. It's structural and chemical characterization of active ingredients, characterization of degradation products. We need to know how many of this product and how toxic each of them. Degradation kinetics important, structural actually questions, conformational changes if it's biopharmaceutical. Of course, direct stability. Most important also polymorphism. Drug polymorphism is a very important issue. For example, on this slide I present to you one actually very interesting example when we just talk about affinity. You know what this is, therapeutic toxicity index. This actually index which show that you cannot exceed some concentration of drug, otherwise it becomes lethal. Yeah, for example, and we know that most, like 90% of drugs, yeah, when you inject it, they get into bloodstream. That first, they're not going to target, but they bind with two proteins. One of them albumin, and another alpha-1 acid glycoprotein. They bind with this protein, and we know that albumin, serum blood albumin, human albumin, it's like can bind like 140 or 160 different ligands. It's like cleaner in our organism. It's one of the abundant protein in our bloodstream. Then it will be immunoglobulins. Yeah. So they first bind, all drugs binds to the to this protein and then distribute it to the target. Imagine now that binding property change under space flight conditions. They certainly change because electrolyte level change in bloodstream and a lot of receptor change, whatever. We know this, but we don't know how exactly. Imagine, for example, and we just measure binding property directly, for example, using different analytical kind of instrumentation. We can say about affinity. We can say that how we should change dose of drugs. Take into account space flight environment. In this, for example, flight I present immunoglobulin, which we tested, which bind analgesic drug ibuprofen. And the left side will present direct measurement stability using differential scanning calorimetry, for example, or it could be another direct technique which address a question of physical stability. And the right side, it's molecule of albumin, which red and blue shows different kind of distribution of electrostatic actually charges, positive in the red and negative in the blue. And of course, electrostatic interactions, it's very important for drug binding. Yeah, and if changes in electrolytes in the bloodstream during space flight change, of course, electrostatic interactions will change. Of course, we know that the hydrophobic interactions also contribute to this. We're using, for example, in ibuprofen, we just identified that two forms. By the way, one form is actually active, S form, and another R form not active. But in organism, there is special enzyme which allow to transfer one form to another, from inactive to active. And 3 months ago, suddenly it appears very interesting article, a research article, which shows that this form, which was not active, and we thought that absolutely useless, has a high potency to prevent development of cancer. So this means that they found that in a cancer cell, this enzyme which transforms ibuprofen from one state to another, level of this enzyme increased dramatically. So if you take ibuprofen, enzymes start to work for conversion of ibuprofen instead of helping cancer cell develop, and so you can mitigate cancer development. So in this case, I will show just one analytical instrument, which we use also for our evaluation of drug stability or potency, so-called Raman spectroscopy. Together with force microscopy, it's allowing you to monitor, as you can see in different color, different components of formulation of these drugs. And on the left, you can see spectrum of Raman spectrum, which in red, blue, and purple shows you spectrum for different components. So for one scan of beam, laser beam, yeah, you can just immediately identify a state of different components and say whether they already degraded, whether how they change during flight, how they interact between each other. It's very useful actually technique which we're currently using. And of course, we just kind of monitor degradation, just normal degradation during storage. You can see on the top of this slide at pH 7 and its stomach pH, which is very acidic, pH 2, you can see that a degradation drug over time is completely different. Of course, we just tested in direct efficacy of potency of amoxicillin, which is one of the very wide actually used antibiotic using microbial culture. And you can see on the bottom MIC data. MIC stands for minimum inhibitory concentration. This is concentration which minimum inhibitory concentration would inhibit, for example, development of bacteria. And we tested under different pH, under different exposure and radiation, which mimic space flight environment. And we can say how, first of all, potency drugs change by flight conditions or this space flight factor. And secondly, of course, we can make judgment working with bacteria which already was in space, how pathogenicity of bacteria change. That's very important because we know that it's increased very, very substantial. Now, when we were talking about pharmaceuticals, I should note one thing. I mentioned one thing, that all pharmaceuticals, all drugs which we used in International Space Station over 2 decades, it was mostly pharmaceuticals in solid formulation like pills. But at the same time, we know that most potent drugs against cancer prevention, for example, radiation protection, immunomodulators, it's biopharmaceuticals. And when we're talking about biopharmaceuticals, it's a completely different issue of stability. You know, this biopharmaceutical biological macromolecules. It's mostly proteins, DNA, RNA, and they're more sensitive to radiation, for example, to any space factor because their activity related to conformational stability, to unique protein conformation which only active and unique for such a certain polypeptide chain. So, and we're working now with pharmaceutical, and just one thing, because I mentioned about Amgen, which developed so-called sclerostin inhibitor. And this sclerostin inhibitor is just antibody. It's immunoglobulin. And immunoglobulin in our blood, you know, but most important, it's very complex protein which has many domains which have different sensitivity. Of course, for example, pharmaceutical companies using this immunoglobulin now very widely. It's one of the potent drugs, and you can see on the bottom that all of this market is just unbelievable. $167 billion 2015. And want we or not, but finally NASA will, of course, consider to use biopharmaceuticals for space flight, especially to protect against such kind of dangerous factors as bone loss and radiation. And last one, which I want to address, which never actually was addressed at NASA, adverse drug effect on astronauts. And we're talking about genetic polymorphism. We know that all people are different, right? And we have some kind of, and now individual medicine or personal medicine becomes a very kind of rapidly developed area of research. You know that if one drug can help for one person, it could be completely actually differently affect another person. And this is due genetic variability. And in this kind of slide, I present a kind of standard dose which you apply for different genotypes. And of course, on the background areas, you know that if you have some kind of mutation in one gene, for example, which is responsible, for example, for utilization or metabolism of drugs, and this is very common, because for metabolism of drug responsible our liver and kidney, right? And we have so-called cytochrome P450 system of enzymes which metabolize different drugs. And these enzymes have different mutation, and different people have different mutations. Let's suppose we have 17 such kind of enzymes. Probabilities that one person have one mutation as presented here in chromosome immediately will affect therapeutic efficacy of drug and, of course, adverse drug effect. So this is important issue. Why? Because here just presented how it's important, this genetic variability. What you can see, small intention, oral administration of drug, it's coming through portal vein and getting to liver. And in liver you have a number of enzymes. They call it with different, like here, CYP3A, whatever, which metabolize different drugs. And on the bottom, on the right, you can see a diagram which show that it's not many of such enzymes. And this is how important that, these enzymes or their contribution to drug metabolism. And you can see, for example, one enzyme, CYP3A, very important. Actually, it metabolizes most of the drug. And another kind of with some contribution. Of course, it's enzymes, it's proteins with the unique structure, which actually mutation can change conformation of proteins, and interactions with the receptor and functional activity will change. And this is actually an important issue, because when you're just taking drug as presented on the top right, yeah, of course, genes control drug absorption. And for example, what I talked to you about human serum albumin, this protein is important. And I told that it's 90% of drug binds to this protein as soon as it's in a bloodstream. But imagine now that you have mutations. Someone has mutation in a serum albumin which change binding property of this drug. Of course, absorption of drug and distribution will be different. And of course, in the next one, when we're coming, not already consider pharmacokinetics, but pharmacodynamics, when drug interact directly with target, of course, it's also controlled by genes. And depending what kind of genetics you have a different contribution from beneficial effect and adverse drug effect. This is important issue. Why? Because so far, no one from astronaut actually was let's just put in such case. Astronauts is federal employees, right? And they have some right not provide information or private information to public. So you cannot ask astronauts to give their blood unless they volunteer. So you cannot test their genetic variability or this, because now we have a facility at NASA Ames Center, for example, which allows you to obtain information about whole genome. It's not only your genes, but also bacteria genes, viral genes, which people have currently, many other genes, yeah? And we have such kind of capability. But unfortunately, we cannot say that, okay, before flight, please give us actually a drop of blood or buccal cell from your cheek, and we can analyze and say whether you should use or not use some medication. So we believe that soon, because it's ethical issue, an ethical issue, it's very important. We believe that soon we will kind of address this for NASA, because pharmacology actually one of the major actually approach to keep astronauts healthy, because deviation from normal physiological conditions we already know exist. Now only question how we can mitigate and handle this. So I believe that, of course, it's kind of too much details maybe, but I give you just kind of general impression where we are approximately and what is concern us. For example, in my case, I'm very really concerned about that NASA age, NASA employee, scientists should be dropped significantly. I mean, we need fresh blood. We need younger scientists which would motivated with this. We estimate that yes, frustration of public sometimes exists regarding that it's slow development in the space exploration. That they annoyed that 40 years, for example, 50 years, we just around the globe, not going, of course. But you should understand that this is very important issue. We collect information. We're using International Space Station's unique laboratory, which allow us to accumulate so much data, which we can predict with some kind of factor or risk, whether we're going to Mars or not going before sacrifice an astronaut. Next, I will show just a couple of photographs from my laboratory. We're working in my group, working addressing some kind of implant in mice and rats. And this is, guys, happy hours, I don't remember, in working. And this is actually we during actually tissue sharing for animals in the laboratory. And this is Halloween party in our laboratory. We're just kind of trying to get some kind of hours to enjoy life. So thank you very much again. If you have some question, I would be happy to answer them.